In my family, my brother and I both have glasses. But both my parents don't have glasses, so I always thought, then why do we? And then how on earth can we say that my brother and I inherited a refractive error from them? I pondered this question of, since we inherit genes from our parents, how did the condition show up in me and my brother? Until I found out in my 7th grade science class, where we had our first lecture on Mendelian inheritance of traits.
In the 1860s, an Austrian monk named Gregor Mendel performed his famous pea plant experiments. He crossed short pea plants and tall pea plants 30,000 times over a course of 8 years. He observed that all of the offspring were tall plants. Let's see how. Each plant has 2 traits that determine height. Let uppercase T stand for the dominant trait. Then we have lowercase t for the recessive short trait. What this means is that it only takes one capital T to make a tall plant. However, it takes 2 lowercase t's to make a short plant. When he first crossed tall and short pea plants, the short trait factor seemed to disappear. However, when he kept on crossing, somehow the short trait factor appears again. Mendel called the tall height trait the dominant trait since it covered up the short height trait. And then he called the short height trait as the recessive factor since it was hidden by the dominant trait. After doing years of research, he was able to conclude that approximately 1 out of every 4 offspring will be a short plant. As I will explain in this example, Mendel crossed a homozygous dominant tall parent with a homozygous short parent, small t, small t. And here is how the 19th century scientist Reginald Punnett structured Mendel's work so that it could be used to predict future outcomes of traits. He created a 2x2 grid, which on the x-axis, he put the tall parent, and on the y-axis, he put the short parent. And this is the tall parent. And the alleles are capital T, and this is the short parent, with the alleles being small t, or lowercase. And these are the alleles. These are also alleles. And when he actually crossed these 2 parents, first what you have to do is bring this one down, like that. So you have a trait from the dominant, and then you have the other. Therefore, as we discussed before, even if you have one capital T, or as we say dominant, even if you have one dominant in your genotype of the final offspring, it will definitely take over and be tall. So, therefore, 100% of the offspring will be tall. But now, this is between a cross of the homozygous tall parent and the homozygous short parent. But what if we take a cross between the 2 of these offspring that it created? So what if we take the cross between this and this? Let's see what offsprings it will give us. So as we saw before, we had one from here, one from here. So it's like this. Then we have the second one. So let me write all the genotypes really quickly over here. We have capital T, then capital T, lowercase t, capital T, lowercase t, and then small t. And what this means is that this is short, and all of these are tall. So we can say that 75% are tall and 25% are short. So therefore, 1 in every 4 of these offspring will be short. Now let's look at the Punnett square. This is my mom. She does not wear glasses, but she has a mild farsightedness. So she has 1 uppercase E and 1 lowercase e. My dad, he has normal vision, so he has both lowercase e's. And when we use the Punnett square, we realize that there's a 50% chance of me having farsightedness.
So now you may be thinking, why should we care? Eye conditions and many other diseases are molded by genetic and environmental factors. By using these techniques, we can predict and manage early enough to prevent further worsening of these conditions. We can improve so many lives just by being able to predict the chance of condition by knowing the genetic history of your parents.